Our next early career investigator, Dr. Nika Shakiba from the University of British Columbia, explores how stem cells compete, cooperate, and evolve within developing tissues. Her work blends bioengineering and developmental biology to uncover the rules of how cells decide who thrives and who dies, insights that could transform regenerative medicine. Please welcome Dr. Nika Shakiba speaking on the molecular rules of embryonic stem cell competition.
Well, first of all, I just want to say it's a great honor to be speaking on the Gairdner stage, and I really want to thank the Gairdner laureates, Drs. Greenwald, Struhl, and Artavanis-Tsakonas, who chose me to be the ECI speaker in their session. I'm an engineer by training, and although my lab is a stem cell engineering lab, and we're motivated by the idea that you can turn stem cells into engineered entities for regenerative medicine, our core belief is that in order to do that, we have to spend our time to first uncover the molecular rules that determine how the stem cells behave. And today I'm going to tell you a story in that vein, understanding the molecular rules of competition between our cells. And this is a story that's led by a very talented Ph.D. student, one of my first two students, Kieran. So if you think back to your origins, perhaps you can't remember that far back, but we all started from a single cell, the fertilized egg. And this cell has the remarkable job to go through a series of very well-defined divisions to create first this cluster of cells, this blastocyst, that then has to coordinate together. This group of cells, this society, has to coordinate to make our tissues and organs and ultimately build our bodies. And the way that this is done, well, these cells have to communicate with each other, and we heard a lot about notch signaling. So these kinds of fundamental mechanisms that cells use to communicate with each other are required for that coordination. But what's interesting to my lab is that sometimes it's not a coordinated effort. Sometimes there's riots. And there's competition between the cells as they're building our bodies. And this is a relatively newly discovered cellular phenomenon. It was first reported in 1975, this idea of cell competition in the Drosophila. And what these studies did is they derived these minute mutants. So these are ribosomal protein heterozygous cells that can fully form a fruit fly. So in the embryonic development, they have no trouble creating a viable fruit fly. The fruit flies are largely normal. They have slightly shorter bristles, as you'll see, but they're functional. But when you mix these minute mutants with wild-type cells, normal cells, and you allow that fruit fly to grow, what you'll find is that the winner cells, the wild-type cells, will actively eliminate the minute mutants from the embryo. They seem to do this in a context-dependent way, so the losers only get eliminated if winners are nearby. And it seems to be that this is contact-mediated or proximity-mediated, so the winner needs to be near the loser to do that. Now we know that through more recent examples, this is a conserved mechanism. So for example, when the zebrafish is being developed, cells that are in the tissue patterning process and receiving slightly higher or lower levels of Wnt signaling will get eliminated by their neighbors in order to make sure that our tissues pattern correctly. And now we know most recent examples in humans. So if you take embryonic stem cells from the embryo of humans and you grow them in vitro, you'll find that certain genetic variants, which are those in green, can actually actively eliminate loser cells, the wild-type normal cells from culture. And this is an active, again, contact-mediated killing mechanism. In one of my favorite examples, a lab, Jinwoo's lab in the US, actually did this competition arena where they took embryonic stem cells of different species and they decided to compete them against one another in vitro. And what they found was that, again, this is a conserved mechanism that even across species you can have competition. And they showed that, for example, if you take mouse or rat cells and compete them against human or rhesus, they'll eliminate human or rhesus. And unfortunately, the cow is the ultimate loser. So the best example that's understood in cell competition in the context of mammalian systems is in the mouse. And we know from looking at mouse embryonic development that competition between cells is conserved across developmental time points. So in that wall of cells I mentioned, the blastocyst, the cells that are taking slightly too long to make their early life decisions, what kind of cell to become, primitive endoderm or epiblast, those cells get actively eliminated by their neighbors or that are saying, hey, you're taking too long, the embryo needs to develop, we need to get rid of you in order to progress. And then after that embryo implants into the uterine wall and starts to develop into the body, at that stage of development, about a third of the cells inside the embryo that are responsible for making the mouse get eliminated. A third of the cells. That's pretty remarkable. And we now know that we can actually engineer those winners and losers. So for example, a study has shown that you could perturb the levels of MYC, which you might recognize as an oncoprotein. And if you have slightly higher levels of MYC, that will make you a winner and you will completely take over the mouse at that stage. Later on, as our tissues are forming, for example, as the skin is developing, you see, again, examples of cell competition that help to make sure the skin's architecture develops correctly. What's good and handy for my lab is that actually you can take, as I mentioned, those embryonic stem cells out from that early stage of embryonic development, put them in the dish, and these so-called mouse embryonic stem cells retain this competitive phenotype. And we now have this kind of hallmark assay of cell competition where you can actually take genetically different mouse embryonic stem cell lines, so they're on the same genetic background, but they have perturbations to key genes involved in cell competition, and you can paint them with fluorescent proteins, different colors, and then the competition phenotype is seen when you compare what happens when these competitors are grown on their own, where you'll see that they grow normally, they're quite viable, but only when you mix them in co-culture, you see that losers are eliminated. So again, it's this context-dependent death that defines competition. Now, using these in vitro and in vivo systems, we've learned some things from mouse embryonic stem cell competition. Well, first of all, we know that there's certain genes, some oncogenes you might recognize, like P53 and MYC, that if you perturb, you can make a winner or a loser. So you can make winners by knocking out P53, you can make winners by overexpressing MYC. And mTOR activity seems to also be important. So higher mTOR makes you a winner. But all of these seem to be sufficient, not necessarily necessary, I should say, for competition. And this led my lab to ask, well, what is the fundamental necessary molecular node to make mouse embryonic stem cell competition happen? Now, it turns out we don't have to fly blind. Because this is conserved, we can turn to really robust data from the fruit fly literature that has shown us a lot about the role of the ribosome in the context of competition there. As I showed you, minute mutants have ribosomal protein perturbations, right? So you lose a copy of a ribosomal protein and you become a loser. And since then, studies have shown that really any or many ribosomal proteins, if you lose a copy, you're a loser. But perhaps what's more interesting is that what's necessary for a loser to die in competition in fruit fly is the ribosome. So losers, as they're dying, they will activate XRP1, which is a key transcription factor that then triggers the stress response in these cells, the integrated stress response, and this ultimately shuts down translation. It shuts down the ribosome through EIF2 alpha phosphorylation. And so looking at the Drosophila literature, we hypothesized, well, maybe the ribosome is also the key node, the missing node that we haven't been looking at yet in mouse embryonic stem cell competition. And so Kieran turned to our collaborator, Tristan Rodriguez, who generously shared with us mouse embryonic stem cell lines that are known winners and losers. And so we took a few of these lines. So for example, we took wild-type cells and BMP receptor 1A knockout cells. BMP receptor 1A knockout cells are actually losers when you mix them with wild-type. And P53, as I mentioned, if you knock that out, that's a super competitor. That can actually eliminate the wild-type cells. So we had these two competition assays where in one case, wild-type cells are losers, and in another, losers, and in the other, they are winners. And we recapitulated these cell competition assays as we expected to see. We showed that when you grow the cells on their own, they're quite viable, they're pretty normal, but then in co-culture, you start to see that the winner overtakes in both situations. And this is perhaps more clearly shown when you look at the ratios of the cells on the right. Now, Kieran said, well, I think the ribosome's involved, so we can't do transcriptomic analysis. And we decided instead to take the cells at this day 3 time point where you start to see competition emerging, and we did proteomic analysis. So we sorted the winners and losers from the co-cultures and the monocultures, and we did mass spectrometry. And we looked to see what's changing. Do we see ribosomal terms? And when we did GSCA analysis on this omics data set, the first thing we noticed is that even in the absence of competition, so before the competitors have even had a chance to compete, it looked like our losers maybe have some level of deficiency in ribosomal biogenesis or assembly. For example, you see that the large ribosomal subunit seems to be dampened in our losers. And then in co-culture, when they're actually competing, you see the losers, the really bright blue here, indicates that they're shutting down ribosomal assembly and biogenesis processes. On the other hand, winners seem to elevate those processes. And so now we asked, well, like Drosophila, is the integrated stress response involved? And so we turned to our collaborator, Ramon Klein-Geltic, who did these Western blots for us, and we actually don't see phosphorylated EIF2 alpha, so we don't think the integrated stress response is involved in mouse embryonic stem cell competition, and our omics analysis actually corroborates this result as well. So not ISR, where is the ribosomal bottleneck then in mouse embryonic stem cells as they're competing? For this, we then turned to Li Vu, speaking of team science, a lot of scientists involved in supporting our work. We turned to Li Vu and we did polysome profiling. And what this allowed us to see is to actually measure the relative quantities of the different parts of the ribosome. So the monosomes, the 40S, the small and large subunits, as well as the fully formed ribosome, the monosomes, as well as the polysomes. So these are multiple ribosomes bound to the same piece of RNA and translating it. And the signal that came out of these results was that we always found that the losers seemed to have a relative deficiency in the large subunit, which is something that also came out from our proteomics analysis. And this persists during competition. Does this result in ribosome heterogeneity? We see winners and losers have different relative levels of ribosomal proteins. I didn't show you the heat maps for these, but they have different levels of individual ribosomal proteins. And maybe there could be different pools of ribosomes that are heterogeneous from one another. And around this time, Maria Barna's lab had shown that actually mouse embryonic stem cells have heterogeneous ribosome pools that seem to translate different pools of transcripts. And we were seeing the same differences in key ribosomal proteins that her lab had shown to be involved in heterogeneity in our data set as well. So we thought, well, maybe that's involved in mouse embryonic stem cell competition. But when we actually pulled out the fractions, the free subunit, monosome, polysome fractions of our polysomes profiling data, and we did mass spec on that, we looked at the ribosomal protein stoichiometry, and we didn't see any difference between competitors before or during competition. So this made us rule out ribosome heterogeneity. That's not involved here either. Okay, great. So does this mean anything? Are the losers actually shutting down translation the same way that happens in Drosophila as they're losing? And so we did an experiment using SILAC labeling. So this is where you basically add amino acids that are heavy labeled into the media. So we did this about 6 hours before we assayed the cells, and then we did mass spectrometry again. And what we found was that losers, before they're competing, so in the absence of competition, they're translating at the same rate. So they're producing nascent peptides or proteins at the same rate as the winner cells. But it's only when they compete you start to see that they're shutting down translation. And both wild-type cells and BMP cells do this when they're losing. And what was interesting to us is that when you actually look at do omics analysis, GSCA analysis on the nascent peptides that the winners and losers are producing, what you see is that actually losers are producing target genes of the MYC pathway, for example, which is a key fitness-associated gene in cell competition. They're expressing actually higher levels of MYC target genes than winners before they're competing. And it's after competition that they start shutting down these fitness-associated genes. So we think that there's some kind of positive feedback loop where losers are then enforcing down regulation of the expression of fitness genes as a result of their ribosome bottlenecks. And so this is now our updated working hypothesis. We've observed that it looks like losers have bottlenecks in their ribosome production. They have fewer ribosomes when they're competing, this limited ribosomal pool, and they start to shut down translation during competition. And so we've now amended our hypothesis to say that because of these ribosomal bottlenecks, this is leading losers to exclude a subset of their transcripts. They're not translating a subset of transcripts. And as a result, this is making them lose. This is the molecular mechanism we think that's involved. And now we're working on assessing this using a synthetic biology technology out of Jean Yeo's lab called ribostamp. And the idea of this technology is they took a ribosomal protein, in this case RPS2, and they fused it to a base editor, APOBEC. And APOBEC basically allows, as that ribosome is assembled and it's translating a transcript, it causes mutations to occur in that mRNA. And so when you do next generation sequencing, you can actually read out not just the abundance of a transcript, but also how much that transcript is being translated, or so-called the translatome. And so we've generated these genetically engineered mouse embryonic stem cell competition lines. With this ribostamp, we've confirmed that ribostamp's expressed. The cells look like, at least transcriptionally, they're normal. They still compete the normal way. And when we look at ribostamp editing, we see that they're successfully editing above the baseline levels in the coding sequence in the 3' UTR, which is where you would expect to see it. And we've now just recently, hot off the press, done this single-cell RNA-seq time course run where we've allowed our competitors to compete with each other and assay them every 12 hours in competition. As I speak, Kieran is working on processing this data and looking for the edits to the transcripts to see how the translatome's changing in competition, so stay tuned for those results. I just want to loop back to the kind of central vision of my lab, which is not a sole vision of my lab, but we're really wanting to understand how interactions, competition, cooperation between embryonic stem cells and pluripotent stem cells shapes how our bodies are built, and we fundamentally believe that we have to unlock these rules in order to port them over to stem cell engineering to make cell therapy manufacturing processes more robust. And the best way to do this, we think, is through team science. So in a recent international consortium that I co-founded with Drs. Maria Abushakra and Nozomu Yashi, actually Maria's in the audience with her daughter there, we created this international consortium around the central vision that you could bring computational biologists, mathematical biologists, empirical scientists in developmental biology and stem cell research around the central vision of distilling these rules of development together and making a virtual simulator of human embryonic development that will help us do that. And by doing that, then we will really understand how our bodies are built. Then we can really engineer cells for therapeutic purposes. And I just want to end by acknowledging the people behind the science. My team has quite grown in the last few years, and I'm really privileged to work with these really bright minds that make my day-to-day life fun. And I really want to thank the funders who enabled us to continue tinkering in the lab over the years. Thanks so much for your attention.